the Immune Zoom seminar tonight. Hope everyone on the East Coast, especially the Northeast, stay dry. Henry is coming. So it's my great honor today to introduce Zihai Li, a renowned medical oncologist and immunologist. Zihai obtained his MD from Zhengzhou University College of Medicine, and then pursued his PhD from Incheon School of Medicine at Mount Sinai. And after PhD, I think Zihai really took on a challenge that deterred many of us. He went to do a clinical residency in Albert Einstein in New York City, and then a fellowship at Frederick Henderson Cancer Research Centers at University of Washington. And then he became assistant professor and then promoted to associate professor with tenure and in the Department of Immunology in the University of Connecticut Health Center. In 2010, he moved to Medical University of South Carolina as a professor and attending physicians. And during his time in South Carolina, I think Zihai really set a great example for many of the young scientists as someone who can really break the so-called glass ceilings for immigrant scientists. Zihai became the leader for cancer immunology program there, and also became the chair for the Department of Microbiology and Immunology. Just 2 years ago, in 2019, Zihai moved his lab to Ohio State University. Not only is he leading the effort in cancer immunology there, he's also built a whole new institute with endowment of $65 million to build the Plantonio Institute for Immuno Oncology. I think this is a great opportunity for him to achieve his vision, and together with many of colleagues at Ohio State, looking for cures for cancers. Zihai has made numerous seminal contributions to immunology and cancer immunology. For example, Zihai's team has demonstrated that the important role of H-cell proteins and the homologs in regulating both innate and adaptive immunity. And particularly, he pioneered in translating his bench-side settings into immunotherapy for human leukemia. He also served on numerous committees and won numerous awards during his career. That's really challenging to list them all during these short introductions. So without much further ado, let's welcome Zihai. Zihai. We're super excited you can be here and look forward to your seminar.
Sure. Thanks so much for that really very generous introduction, and very pleased to be here, and I really want to thank the organizers to invite me and really put together a nice forum to discuss common interests in immunology. So let me see I hope you all can see my slides. So as Shu just mentioned, I'm now here in Columbus, Ohio. Ohio, as you know, is huge. Columbus, specifically a huge college town. When you say OH, everybody yell IO. Well, IO, in my opinion, stands for Immuno Oncology. This is what we do, but today I will actually show you one line of work my lab has conducted over the years related to the role of platelets in immuno-oncology. And certainly platelets are known to be important in control hemostasis, but what does platelet have to do with immuno-oncology? I hope to impress upon you and with this line of work we have done in recent years. This is my disclosure. Is really not relevant to today's discussion. I don't really need to highlight the long journey we collectively have embarked on in cancer immunology. Obviously, this has been going on for many years, many decades. From the early observation of presence of immune cells in tumor, the practice of injecting bacteria to sarcoma patients for treatment of cancer pioneered by William Coley. And of course, the concept of tumor immunology really was proposed by Thomas Burnett and so forth in a concept called immune surveillance. And this was followed by experimental validation of tumor immunity. Arguably, one type of clinical practice, i.e. bone marrow transplant, is actually one form of immunotherapy. Granted, this is in allogeneic setting. And of course, the rapid identification of tumor antigens, tumor-specific antigens, tumor-infiltrating T cells, the proof of cytokines for immunotherapy of cancer in type 1 interferon IL-2, and then the work from Bob Schreiber and others demonstrating there is an active immune editing process during oncogenesis, and arguably one of the strongest evidence to support immunology, of course, is the immune tolerance mechanism actively elicited by tumor to the host. Of course, rapid forward, we now in the era of clinical immuno-oncology with a variety of immune checkpoint blockers for treatment of not only melanoma, but a variety of solid tumors, PD-1, PD-L1 blockade, oncolytic virus, and of course, chimeric antigen receptor transduced T cells targeting CD19. I actually did not list BCMA. Of course, CAR has been approved now for treatment of multiple myeloma. Now the question is, where we're going? Where are we going from this point on? And of course, the glass is still half full. We know only 20 to 30% of patients respond to checkpoint blockade therapy. How do we overcome both primary and secondary PD-1 resistance? How do we broaden cell therapy beyond the treatment of hematologic malignancy, and how to reduce or eliminate immune-related adverse events? All those things are challenges. Okay, let me see. All right. So immunotherapy now considers one of the pillars of cancer medicine. A new pillar of cancer medicine represent a new paradigm. Of course, it is treating the immune system, not the cancer. And many of us believe, and it is really one of the only modality that can offer a cancer cure because of the specificity, adaptivity, and of course, memory inherited in adaptive immunity, and of course, it's tissue agnostic. One of the key challenges, simply put, is indeed to overcome resistance, and that is convert resistance to a sensitive tumor. And I think beyond PD-1, PD-L1, and CAR-T, so what is the next big thing in immuno-oncology? I think, obviously, if you look at the immune system, the immune responses, and especially what is happening in the tumor environment, a lot of switches and buttons you can push, but which one of those switches are the key? This is nicely illustrated by this review article in Journal of Cell Science, and I don't have to discuss what are the complex components, cellular components, molecular components in the tumor microenvironment. But the question is, what is really missing? One thing I want to argue, at least in this particular talk, is actually the abundant presence of platelets in the tumor microenvironment. Once again, the question is, what has got to do with cancer immunity? If you survey the literature, interestingly, thrombocytosis has been linked to poor outcome in a variety of cancer types, especially ovarian cancer. A paper published by a new suit in 2012. It was further shown by many folks that the platelet-derived TGF-beta can actually contribute to EMT and promote metastasis. If you're interested in this topic, and I think this is a very nice book called Platelets. The fourth edition published in 2019, we contributed to a chapter specifically discuss the role of platelets in tumor immunity and immune evasion. In that review, we highlight some of the epidemiological evidence that thrombocytosis really contribute to cancer poor outcome. This is true in colorectal cancer, ovarian cancer, non-small cell lung cancer, gastric cancer, basically you limit thrombocytosis in cancer confers poor prognosis. Okay, so the key unanswered question is what are the roles and the mechanism of platelets in shaping anti-tumor immunity? My lab has been working on this molecule called GP96, otherwise also known as GRP94, as Shu mentioned in the introduction. And this is an endoplasmic reticulum paralog of HPV90. It is played a very important role in many aspects of development and immune responses. Especially, we demonstrate that the GP96 is a master chaperone for toll-like receptors. This was uncovered almost 20 years ago. But accidentally, we also found that GRP94 or GP96 is important for folding a receptor, a glycoprotein 1b95 complex on platelets. So this is a platelet-specific molecule. It's critically important for platelet adhesion, activation, and coagulation. And what we found is that if you knock out GP96 from platelets using PF4 system, you have essentially a model called Blast-Smear syndrome. Basically, it's a macro thrombocytopenia with defect of GP1b95 complex. This complex, also known as the von Wernberg factor receptor, VWF receptor. The bottom line is that this is very critical for the function of platelets. Okay. All right. So sorry, somehow my slide stuck on that particular slide. So the question, we try to answer a number of questions utilizing this platelet-specific GP96 knockout model, which is essentially basically the platelet dysfunction model. So the first question is, do platelets play any role in immune surveillance? To answer this question, we subject both wild type and knockout mice to carcinogen called mescalenthrin MCA. It's one injection, and followed 19 weeks later by looking at a variety of things. Of course, a tumor growth curve, serum cytokine, and what's going on in the tumor microenvironment. And by the way, this method has been used extensively by folks who study immune surveillance, including Bob Schreiber. And what we found was that the knockout animals actually are better in protecting MCA-induced fibrosarcoma. This indicates as tumor growth curve right here. And we also found using immunofluorescence staining that there are significant more tumor-infiltrating CD4 and CD8 T cells in the knockout MCA fibrosarcoma. And this is correlated with increased functionality of CD8 cells in terms of interferon gamma production. This is done in the draining lymph nodes, but this is also true if you isolate tumor-infiltrating T cells. So then the second question, can platelets affect immunity against transplantable tumor? And this is, of course, relatively easy to do. We basically took wild type and, again, platelet-specific GP96 knockout mice and transplant MCC8 colon cancer. As you can see, once again, knockout mice seem to be better in controlling the tumor growth. Not surprisingly, in the tumor microenvironment, we also see an increased functionality of both CD4 and CD8 T cells in their ability to produce cytokines, including gamma interferon, TNF-alpha, and both. So the third question, can platelets modulate effective functions of T cells? So in this case, we basically utilize a double transfer model, and we implant B60 melanoma into wild type or platelet-specific GP96 knockout animals, followed by lymphodepletion with cyclophosphamide, and then infusion of antigen-specific T cells, in this case, P-male, which are activated ex vivo. And then 24 days later, we sack animals and look at the functionality of those T cells. So the bottom line is that we found, once again, if the host has poor platelet function, in fact, the T cell mediated tumor control is better, indicated in this red tumor growth line. And that, once again, correlate with the increased functionality of this antigen-specific T cells as far as interferon gamma secretion is concerned. So the next question is, do platelets secrete soluble factors to inhibit T cell activation? In this particular experiment, we took essentially platelet-rich plasma isolated from either mouse or human being. We activated with thrombin, a very potent activator for platelet. Then we purify we then isolate the superlatent from this activated platelets, and we incubate with sort of polyclonal T cell activation system, CD3, CD28, along with IL-2 stimulates naive T cells. In this case, T cell labeled with CFSE. So we look at proliferation and cytokine production. The bottom line is that the secretome of active platelet are shown to inhibit T cell response. This was reflected by decreased size, decreased blastogenesis, decreased production of TNF, coenzyme B, gamma interferon, and porphyrin. And then the question is, what are the molecules that involved in suppressing T cell function? And this, to answer this question, my student, Salih Rashidi, actually underwent a biochemical approach by fractionate the secretome of active platelets and then ask which fraction are able to inhibit the T cell activation. So he actually found there are two fractions, fraction A and fraction B. Okay, and it turns out the fraction B is a lactic acid. I'm not going to talk too much about lactic acid. Fraction A, however, is a TGF beta. And this was demonstrated by mass spec of isolated fraction. And he also showed functionally in this suppression assay, if you block TGF beta, and the fraction essentially lose the activity of suppression. This is what's shown, was published in this 2017 paper. So of course, what is TGF beta? This is a main character of this story. I just want to mention briefly. TGF beta, of course, made as proform in a secretory pathway and went cleavage by furin-mediated mechanism, and then it folded as this latent TGF beta complex, mature TGF beta node. Noted here is form a disulfide bond. This is a dimer, but this form is not active because it's wrapped around by this latency-associated peptide called LAP. So this complex needs to be activated in order to release active TGF beta. Variety of mechanism to activate TGF beta. This is showing here. So this latent TGF beta form can exist as a soluble form in a plasma. It can also bind to large TGF beta binding protein, which is associated with extracellular matrix. I will tell you more about this particular molecule. GOB is a type 1 transmembrane protein expressed on cell surface, and this protein can also bind to a latent TGF beta. In any case, all those complexes need to be activated to release active TGF beta. One of the mechanisms is well worked out is alpha V beta 6 beta 8 dependent mechanism. Okay, why do we care about TGF beta? Again, I don't have to talk about this. Obviously, TGF beta 1 does a lot of things, but it's really a master cytokine in regulating inflammation and tolerance. And genetically, it was demonstrated very clearly. If you knock out TGF beta 1, the animals died of multifocal inflammatory diseases. And of course, this is immunological mediated because the disease can be rescued if you cross the TGF beta 1 knockout animals with animals that don't have adaptive immunity. And what is also important is that TGF-beta is a key mechanism for conferring PD-1 resistance. Many people have demonstrated this fact. I'm just mentioning 2 papers published in 2018. One is in bladder cancer by a Genentech group. The other is colorectal cancer. In both cases, they show that if you block TGF-beta signaling, you can convert PD-1 resistance to PD-1 sensitivity. I mentioned the GARP molecule. Now the question is, what is GARP? When I mention GARP, a lot of people say, "Oh, are you talking about this movie? Robin Williams movie, The World According to GARP." It is an interesting movie, but in this particular story, GARP is a glycoprotein, a predominant repetition predominant protein. It's a type 1 transmembrane protein expressed on plasma membrane of regular T cells, tumor cells, platelets, and some activated B cells. And it's encoded by this gene called LRC32 in humans, residing in long arm of chromosome 11, which is actually a region that's frequently amplified in patients with triple-negative breast cancer. But the key is that GARP is a surface docking receptor for latent TGF-beta, and it's very important in activating latent TGF-beta on the cell surface. Okay, so once again, just to show you that the potentially GARP latent TGF-beta complex can be activated. This was shown alpha V beta 6 beta 8 mediated mechanism of activation by Steve Nishimura at UCSF. Elegant work was done last year, published in Tcell. We published earlier last year that there is also a mechanism of proteolysis mediated by thrombin that result in release of active TGF-beta, which I will talk a little bit more later. So I mentioned the platelets seem to be very important in suppress T cell response, and I said that active platelets secrete TGF-beta, and I mentioned that platelets express GARP. The question is, what do platelets contribute to active TGF-beta in the tumor microenvironment? And it turns out a lot. So platelets are actually a dominant source of active TGF-beta, and this is through GARP-mediated mechanism. And there are many ways you can show this. One simple way to do this is just to inoculate tumors, in this case, MC38 tumor, into wild-type animal or platelet-specific GARP knockout animal. And then you look at TGF-beta activity by look at phospho-SMAD3 by IHC. You can see abundant TGF-beta activity in wild-type animals. But if you look at tumors in a platelet-specific GARP knockout, you see very significant reduction of TGF-beta activity, which is very striking. So then the question we ask next is, can GARP be targeted for cancer immunotherapy? And to answer this question, we generate a whole bunch of monoclonal antibodies against GARP. One such antibody is called 43, and this particular antibody is very interesting and appears to completely block the binding of GARP to latent TGF-beta. So to demonstrate this, we generate a GARP, a stable GARP expression cells, which we then incubate with human latent TGF-beta. Okay, in the presence of either isotype control antibody or 43 antibody, followed by cell surface staining of human latent TGF-beta. So if you do the experiment in the presence of isotype control antibody, you see very nice binding of latent TGF-beta to GARP expression cells. But if you do that with, in the presence of 43 antibody, you see completed abrogation of the binding. We also showed that this antibody has mono-agent activity in the treatment of preclinical triple-negative breast cancer model 41 in decreasing metastasis. And we further show that the anti-GARP antibody 43 can synergize with anti-PD-1 antibody for treatment of 41. And this was done using schema as shown on this slide. The bottom line is that we show in this particular model that the combination, that is PD-1 antibody plus anti-GABA antibody 43, leads to 50% of animals to have a prolonged survival beyond the 300 days. And in this particular experiment, mono agent 43 or PD-1 antibody really don't have activity. Most importantly, we show that 43 antibody treatment is to reduction of TGF-beta activity, evidenced by, once again, reduced phosphoSMAD3, and also reduction of alpha muscle actin, which is to be expected when TGF-beta pathway is blocked. Of course, anti-PD-1 antibody does not do anything to TGF-beta activity. Okay, now the question is, can anti-GABA antibody have any single agent activity in GABA negative tumors? To answer this question, we generate a human GABA knock-in animal. So bottom line is that we just replace the extracellular domain of mouse GABA with human GABA, and retain intracellular domain, which is pretty short and doesn't really have a signaling motif anyway. Once we have this human GABA knocking animals, we inoculate murine cancer, bladder cancer, MB49, and melanoma, B16. And then we treat it with a single agent 43, 200 microgram per dose every 3 days. As you can see, 43 does seem to have some activity against MB49, as well as B16. And of course, we want to know, does 43 reduce TGF-beta activity in the tumor microenvironment? I showed you RHC before, but we also showed you using intracellular stain of phospho-SMAD. So as you can see, we found that 43 can inhibit SMAD signaling, TGF-beta signaling, evidenced by the reduction of phospho-SMAD 2 and 3 in every cell that we isolate from the tumor microenvironment, including CD8, CD4 B cells, macrophage, and dendritic cells. So then the next question, can platelet agent, antiplatelet agent pharmacologically promote cancer immunotherapy? So of course, due to researchers in platelet biology, especially folks who are interested in cardiovascular medicine research, we have lots of platelet inhibitors, the old-fashioned aspirin and clopidogrel, which inhibits the ADP-P2Y1 receptor. So once again, we use adoptive T-cell therapy model to address this question. So in this case, wild-type recipient was given B16 melanoma. Then when the tumor established, we gave these animals antiplatelet agents, in this case, aspirin, clopidogrel. Then we did lymphodepletion, followed by transfer of antigen-specific T cells. So if you do this experiment with just anti-platelet agents and radiation or cyclophosphamide for lymphodepletion, you really see absolutely no difference. So it doesn't matter whether you have antiplatelet agent or not. However, if you do this experiment in an adoptive T-cell transfer setting, as you can see, the animals treated with antiplatelet agents enjoy cancer-free survival far better than the animals that did not get antiplatelet agents. And once again, this translates into longer survival and increased functionality of antigen-specific T cells. And then the question is, are there any therapeutic benefit of combination therapy of PD-1 blockade and platelet inhibitor? In this particular experiment, we used MC38. We actually did anti-platelet agent priming. That is, after tumor established, we'll give antiplatelet agents, followed by combination of antiplatelet agents and PD-1 therapy. When you do this, as you can see, the combination therapy is better than PD-1 alone. Antiplatelet therapy alone really does not do anything to MC38 tumor. So then the next question, can platelet GARP-related TGF-beta access be targeted pharmacologically for immunotherapy? To address this question, we want to understand how latent TGF-beta associated with GARP is actually activated to release the mature TGF-beta. In other words, this particular process. So this complex somehow have to be dissociated, mature TGF-beta got to be released. I mentioned already about the integrin alpha V dependent mechanism. This is a paper from Steve Nishimura. Using cryo-EM, he demonstrated that the cell surface GARP, in this case shown in this structure, horseshoe structure, bind to TGF-beta, and this the pro-domain of latent TGF-beta then can then bind to alpha V beta 8 integrin. And after this interaction, essentially there is a pulling to the opposite direction. I'm not sure if there are any question there. I hear some echo there, but the bottom line is that this mechanism, this binding results in accessibility of mature TGF-beta to then engage the TGF-beta receptor. This, interestingly, TGF-beta, mature TGF-beta doesn't even have to be released from the complex in order to exert its activity. But we are asking whether there's any integrin independent mechanism, and we found that actually GARP indeed can be periodically cleaved from a cell surface. And this work has also been published. I just want to briefly highlight this. So I mentioned already about the molecular chaperone GP96, which is involved in folding von Willebrand factor receptor, as well as the toll-like receptors. But in this particular case, we found that if you overexpress GARP in wild-type cells, that is GP96 positive cells, you actually have the full-length GARP, 75 kD. You also have the two fragments, 50 and 28 kD molecules, which is a stark contrast to the GP96 knockout cells. You only have full-length GARP. We also found in the paper that was published in JCI that GP96 is critically required for the folding of GARP and surface expression. So which leads to our postulates that perhaps GARP can be cleaved on cell surface. Once again, GARP is not present on cell surface of GP96 knockout cells. Yes, indeed, we found that you can isolate GARP components in the medium of the wild-type cells, not GP96 knockout cells. And again, by mass spec, we confirm those fragments are indeed derived from GARP. By protease scan, we found two protease sites in both human and mouse GARP, which are consensus thrombin site, this proline arginine consensus protease cleavage site of thrombin. So, which then raises the question that thrombin perhaps can cleave GARP on the cell surface, which then leads to a release of active TGF-beta. And this is kind of important because I mentioned before, thrombin is a very potent activator of platelets. Okay, and just to make a long story short, and we indeed demonstrate that those two sites are utilized by thrombin to cleave GARP. And this was done by site-derived immunogenesis. If you muted both sites and you have no more GARP cleavage. Importantly, we found thrombin incubation, so treatment of platelets with thrombin actually can lead to increased active TGF-beta from wild-type platelets, but this does not happen if the platelets are knocked out of GARP. So in other words, this increased active TGF-beta is actually mediated by thrombin mediated GARP cleavage. This was true in vitro. This also appears to be true in vivo. This is taking wild-type animals and treated with a thrombin inhibitor called dabigatran. you have a reduction of active TGF beta. And if you, in the wild type animals, if you do this experiment with GABA knockout mice, you see with or without dabigatran, you see no differences in active TGF beta level. So, so therefore, we conclude that thrombin can cleave GAB, then results in a release of active TGF beta. Mechanistically, this is probably very easy to envision. This is the structure that was resolved by Steve Nishimura and also Sophie Lucas in Belgium. And this is the thrombin binding site. So what happens is that after the GAB cleavage and this thermodynamically, this complex will fall apart and mature TGF beta will be released. And we then ask the question, can we inhibit thrombin, therefore see some anti-tumor activity? It is appeared to be indeed the case. This is true, utilize multiple preclinical models, including MSC38. And if you do this experiment in GAB knockout animals, and then you don't see the effect of dabigatran or thrombin inhibitor. And then next question, does thrombin inhibitor remodel the tumor microenvironment? So the answer appears to be yes. So so as you can see, we look at the TGF beta directly, TGF beta 1 IHC, dabigatran treated animal. If you look at the tumor, there's much more reduction of TGF beta in the tumor microenvironment. And now, surprisingly, you see collagen deposition is reduced. Alpha smooth muscle actin is also reduced. And the dabigatran treatment does not really affect infiltration of platelets. This is shown by CD61 IHC. Importantly, using CyTOF, we also see after the thrombin treatment, there's an enrichment of both CD8 T cell and natural killer cells in the tumor microenvironment. And then next question, can thrombin inhibitor promote PD-1 blockers for cancer immunotherapy? Now, at this point, you know the scheme. We essentially treat animals with dabigatran, again, thrombin inhibitor, and the PD-1 antibody. We see much better tumor control, much better survival curve. And this is correlated with reduction of TGF beta in the dabigatran treated group, systemically as well as in the tumor microenvironment. So I think at this point, I want to conclude a few things. One, do platelets play any role in immune surveillance? I think we want to see the answer is yes. Of course, a lot of more experiment we need to do, especially related to the immune editing question. Second, can platelets affect immunity against transplant tumor? I think the answer is a resounding yes. We have done multiple tumor models, 5, 6 different tumor models. The answer appears to be yes. Can platelets modulate the effector function of T cells? Of course, it does happen in vitro. It appears to happen also in vivo. Utilize adopted T cell transfer experiment. And then, do platelets secrete soluble factor inhibit T cell activation and function? The answer is most definitely yes, and we like to believe there are at least 2 factors that contribute to this. One is TGF beta, the other is lactic acid. And then can GAB, which is a molecule that binds to latent TGF beta expressed on the cell surface of platelets, be targeted for cancer immunotherapy? I show you some evidence that antibody that block the binding between GAB and latent TGF beta appear to have anti-tumor activity, either alone or in combination with anti-PD-1 antibody. And then, can anti-platelet agents promote cancer immunotherapy? I think our work with dabigatran and also aspirin clopidogrel appear to also indicate that this question is a affirmative one. And then finally, can platelet GAB TGF beta access be targeted pharmacologically for cancer immunotherapy? I think the thrombin inhibitor experiment indeed demonstrated that appears to be possible. So what is the take-home message? I think GAB latent TGF beta pathway promotes immune tolerance. and oncogenesis. This kind of summary, some of the work we published, we found cancer cells can also express GARB, which promote EMT, cancer emission, and metastasis. We found that the GARB is actually also important for the regulatory T-cell biology. And especially we found that the GARB is important for the production of CD103 by the gut-resident regulatory T cells. And we also show that in some subset of active B cells express GARB transiently. That seems to be important for not only IgA class switch, but also important for gut homeostasis. And finally, I hope I somewhat convince you that the platelet-specific GARB is very important for suppression of both CD8 and CD4 cells, and therefore provide a mechanism for tolerance in immune surveillance. And sorry, my slide got stuck here. Okay, and then another piece of message that platelets, especially GARB, I think are valid targets for cancer immunotherapy. And of course, there are a lot of people who are interested in finding the next big thing in immuno-oncology. I'm not saying GARB is the next big thing, but I think hopefully it will enter into a discussion. Currently, there are actually active clinical trials by AbbVie, for example, to look at the role of anti-GARB antibody to overcome PD-1 resistance. The other thing, of course, there are so many questions, fundamental research critical to provide answers. So I just want to end my talk by mentioning to you that here at Ohio State, we're building a significant, sizable program, Immuno Oncology, what Shu mentioned, Planetary Institute for Immuno Oncology. It's an institute we established a couple of years ago. It's a comprehensive bench to bedside research institute focused on harnessing body's immune system to fight cancer at all levels, of course, from prevention to treatment and survivorship. And actually, altogether, we are very fortunate to receive $102 million investment, and we built we think, a first-class immune monitoring and discovery platform. We still have 40+ faculty slots open at all ranks, including senior investigators to lead some of the flagship programs that we're trying to build, including cell therapy, immunogenomics, systems immuno-oncology, translational immuno-oncology, and cancer immunomicrobiomics. So if anyone interested in joining us, drop me an email or send a note to this. And here's our cancer center, the third largest cancer center in the United States. And once again, I want to thank folks who help with the work I presented without going through each of them one by one. But I obviously cannot do anything without my team. And if you want to know more about PIO, you can check the website. Once again, I hope at some point in the future, when you think about Ohio State, you think about immuno-oncology. So thanks very much. I'll be happy to take any question you might have.
Thank you, Zihai, for the beautiful talk. It's really nice to learn fascinating basic biology and health implication in clinical research, especially immuno-oncology. That's beautiful. So feel free to raise your hand or type a question in the chat box. We can go one by one. Wuchuan, you can go first. Zihai, by the way, sorry to cut down your funding by almost $40 million. Seems like you have done a really amazing job of raising funding the past 2 years.
Very fortunate, and the community is behind us. Hopefully, we'll put the money into good use. I think Tron has a question.
Yeah. So really great talks. I fully enjoy it. So I have actually two questions. First, I'm actually curious about the function of GP96 in platelets functionality. So what's the exactly use? I mean, although you describe a lot of phenotype, I'm still
Yes.
curious machinery, how this molecule work for the platelets.
Great question. I didn't talk too much about the role of GP96. This molecule is actually fascinating. There are lots of clientele. It's in the ER. It's involved in folding assembly of lots of cell surface molecules. I mentioned toll-like receptors, which we published this immunity many years ago, and we also show that it's important for folding of GARB. But what is relevant to your question is the fact that this is important for the assembly and expression of von Wernberg factor receptor, GB1B5 complex. Exact mechanism, which is probably what you're trying to get, is not clear. The protein, of course, will be made, and they do not come together to form the complex. Therefore, they are not stable. They're not going to the surface. And do they then go through the ER-associated degradation pathway? Or what else might be happening in terms of the biochemistry? We don't have more molecular insight. That's the short answer.
So, but in your knockout mice, do you see the abnormality of platelets, for example, activation or accumulation?
Yeah, sorry, that part of the question I did not answer. Yes, actually the first hint that there's something wrong with platelets was indeed there was bleeding, prolonged bleeding time.
Okay.
When my student nicked the tail and tried to do genotype, the mouse was bleeding so much. And then we look at the blood smear, and we see this micro thrombocytopenia, giant platelets. And we then realize, okay, this may represent a Bernard Solier phenotype. Lo and behold, indeed, it looks like this von Wernberg factor receptor is not expressed in the GP96 knockout platelets. So yeah, the function is compromised. Function platelet compromised. There is in prolonged bleeding time. There is problem in adhesion, activation, aggregation.
Okay.
Thankfully, there's no other defect. So the only defect is the platelet compartment, which once again provide a nice model to look at the contribution of platelets in immune response.
So when you, let's say, if you have induced a cutaneous wound healing process, the platelets recruitment will be similar, or it's actually also compromised?
It's a great question. I don't have an answer. I think the same thing can be said about the role of GARB. Does it play any role in tissue homeostasis and wound healing, specifically as you just mentioned? I think probably it's worthwhile to investigate.
So when you see the morphology change on the knockout platelets, if you isolate them in vitro, you also see compromised activation of the platelets in the, yeah?
Correct.
Okay.
Correct.
Cool. So my second question is actually when you actually try to see the function of platelets on the anti-tumor T cells by using the supernatant. I remember you're using the CFSC proliferation. I'm actually curious that do you actually also tested the direct interaction between the platelets and the T cells, whether that's also have effect?
It's a beautiful question. Once again, the platelets T cell conjugate has been observed in a variety of settings, especially during infections. I think there are also recent paper demonstrating that interaction is very important for dictating T cell fate. Furthermore, platelets can also form conjugates with other myeloid cells, and so platelets tend to be sticky. I think in our experiment, we purposefully looking at the soluble factor, and we didn't look at the cell contact dependent mechanism of inhibition. But I think your point is well taken. There must be, yeah.
The other thing actually following this T cell thing is, we know that the platelets also have important role inducing neutrophil recruitment, actually, which also important for the tumor environment. So do you see that too?
Yes.
I mean, let's say in your tumor model, in the knockout animal, the neutrophil recruitment also, I guess, will be different, or you don't see any difference?
Yeah, we in the immune editing, I didn't show you too much data because of in the interest of time. But in terms of the immune surveillance question, we induce cancer in a knockout animal. We look at tumor growth. We look at tumor microenvironment. What is actually interesting, exactly as you said, there are changes in myeloid population. There appears to be more immature myeloid cells, so-called MDSCs, in the tumor, in a wild type, much less so in the knockout animal. And this is actually part of the manuscript that we submit to Cancer Cell. We have lots of questions, and we're still trying to fight the reviewers.
Well, nowadays that's the same everywhere. It's all endless questions. That's not something.
Yeah.
Okay, so you do see that the neutrophil, the myeloid cells get compromised in the tumor in your knockout mice.
Yes, that's
see.
right. Yeah, we in fact exactly, this is one of the research directions that we're pursuing, and the other thing, I'm a little bit hesitating because we don't have a lot of data. That's one thing. The other thing is that it's actually not easy to study. Sometimes especially this conjugates, and is this really artifact? Does this really happen physiologically? And how do you prove it? And so there are some great question. I think we need some clarity for sure.
So actually, the same question following the CD8 and the myeloid cells. So I'm curious that in the tumor phenotype you observed that in your platelets defect mice, do you think that the issue on the platelets caused the tumor difference is because of the T cells or because the myeloid cells?
A lot of myeloid people here, I'm sure.
Yeah.
So so I kind of tends to be a little bit biased, and I think eventually it comes down to T cells, I feel. But it could, but then of course that's the after effect, right? Could the most important thing be the myeloid cells? It's possible. I cannot dogmatically say.
I guess the antibody depletion will be able to address this question. So let's say if you deplete myeloid cells or the CD8 T cell to see whether the defect of which the target cells from the defect.
This is what I'm saying. If you deplete T cell, of course, the immunity is compromised. So T cell are important, but they are important because there are more friendly myeloid environment to educate them to do so. Obviously, that's possible.
Okay, cool. Thank you so much. Thank you.
That's a great question. I should really invite my students to listen to this question.
There's a couple more questions in the chat box. It's funny that Tran texted Q, and now Q has 2 more questions following that.
Okay.
So the questions are: have anyone analyzed the clinical trials of PD-1 therapy and found any evidence that anti-platelet agents improve the efficacy of immunotherapy?
Thank you to Q. Okay, well, thanks again. The question very naturally, yes. We have tried to do this in non-small cell lung cancer setting. It appears to be yes. So anti-platelet agent seem to correlate with more favorable response to checkpoint blockade. This is in advanced non-small cell lung cancer. We have now published this, and the reason is that this is all, retrospective analysis. And so what does it mean, right? So prospectively, we're trying to answer that question by prospective clinical trial. So we currently have an active trial combining checkpoint blockade and antiplatelet therapy in the treatment of head and neck squamous cell carcinoma. And hopefully we'll be able to answer that question.
And then the follow-up question from the same person is compare with cyclophosphamide, would you recommend that antiplatelet agents may have a better Tcell therapy potential?
Very interesting. I think it's fabulous. Beautiful question. I don't know who asked this question. There has been papers regarding platelet dysfunction and changes in microbiome. This question is actually very significant amount of literature in that direction. We unfortunately have not looked into this. The leaky gut situation, if there is a dysfunction of platelets, could you have some sort of subclinical evidence of leaky gut situation, and therefore could that contribute to some sort of inflammatory milieu, therefore better anti-tumor immunity? I think two great hypotheses. We just need to address them.
And then another question from a different person is, will the antiplatelets treatment induce coagulation disorder?
Obviously, that's the worry, right? So especially with cancer patients, they oftentimes have all kinds of other issues in draining catheter for IV infusion, among many other things. So yeah, so if you systemically inhibit platelets, it could contribute to problem in bleeding. But we're trying to find a safe strategy. And one other thing actually we're trying to do, which I don't have data, but some direction that we are pursuing, is to generate the GARP so a specific inhibitor to block the thrombin cleavage of GARP only, not so try to separate the function of platelets in active in TGF beta and the function of platelets in control bleeding. So I'm not sure whether it's possible pharmacologically, but this is the direction we're going.
Cool. And one more question in the chat box is, does leaky gut, in this case, associated with microbial dysfunction, potentially contribute to the platelet-mediated immune suppression because of continuous uncurable injury in the colon? And do you recommend to try antiplatelet agents in tumor patients, especially the one with dysfunctional gut microbiome?
Good question. Once again, as I said before, all of those questions are hypotheses are plausible, and we just need more evidence, experimental evidence to address them.
Cool. I have a couple of questions myself to if no one else has more questions. One is that do you know in the tumor cases, what's the inducer for platelet activation?
Usually collagen, collagen deposition. So as you know, the extracellular matrix and everything is actually, fibronectin, everything else, they can, leads to platelet adhesion and then activation. So, but the other thing is important, of course, the platelet have to extravasate, right? So it's in the blood, it has to get into the tissue. So the earlier question from Chang related to the myeloid platelet conjugate, that's actually has been postulated to be one of the mechanisms how platelet get out to the tissue. and which is piggyback on myeloid cells. And this has been shown to actually be important in forming a metastatic niche. Actually, some beautiful paper out there. So, but I think in general, extracellular matrix, perhaps a vascular injury as cancer invade, right? It cause problem, endothelial cell damage, expose everything, so that leads to platelet activation. But one thing I want to mention to stress, I guess, emphasize, is that platelet activation is the rule rather than exception in almost all solid tumors. Because as you know, solid tumor is a chronically unhealed wound, right? The wound, the tissue damage, the body cannot help it. You're gonna have to plug the hole and activate platelets. So it happens all the time, which is not surprising that clinically, patient with solid tumor can present with DVT, thrombosis, because of clot formation.
Another question, this is a little more selfish question that to have a really interesting asset, and going back to the thing you mentioned about lactic acid producing from the platelet, it's really surprising that platelets produce a lot of lactic acid. Do you know they produce this in baseline, or they have to be really activated to really turn on the metabolism, secreting lactic acid?
You know what? It's very good you ask a selfish question. Well, I'm going to summarize the data and send to you, because we did look at baseline situation. There are changes in lactic acid even at baseline. So we couldn't make too much out of that. So and also, as you know, sometimes your student graduate, nobody else is interested, and so I have a whole bunch of data related to lactic acid.
All right, cool. We can talk more offline.
Yeah.
Yeah, sounds great. Troy has one more question.
Hey, Zihai.
Yes.
Me again. So I just slipped one question in my mind that actually I'm a neuroendocrinology guy. So I'm curious that, the platelet is obviously the one of the largest container for the peripheral serotonin.
Yeah.
So, I mean, in your case, because the thrombosis or the delay of the hemostasis, is that because could be that level of the peripheral serum or plasma serotonin is changed, cause all these issues? Obviously, serotonin also promote the tumor growth, regeneration, and then growth. So I'm actually just curious about this. In your knockout mice, whether the peripheral serotonin actually changes. If that's the case, maybe that's caused the general environment change that your tumor growths have been different phenotype.
Well, so that's the beauty of doing presentation like this. I have absolutely no clue. So okay, that's interesting, and we should
because when you mentioned that your knockout mice have issue with activation, so one of the key activator for the platelets is the serotonin. I mean, it's actually induced the activation of the platelets and then cause all the downstream phenotype. In addition, that the bioavailability in the peripheral of the serotonin directly influence the tumor growth. So if the platelet cannot take up the serotonin, then it messes up a lot of things. So that's just my curiosity,
Yeah, no, that's fascinating. Well, I'm gonna have to look into this, and then of course I have to ask you for help.
No, we are writing. I mean, the reason I'm actually think about this because we're writing a bunch of paper about the serotonin-induced platelets activation. That's why I'm actually just have this thought that because obviously the platelets phenotype actually change dramatically in your mouse. So I'm actually quite certain the peripheral serotonin level probably also different, because they cannot just take up the serotonin that your whole model having changed, including the tumor environment.
Oh gosh, you see this, we traditionally experimentalists trained as a reductionist, right? So we just look at one thing at a time and not ignore everything else. I think thanks for that comment and suggestion. So we'll look into this.
Yeah, sure, absolutely. I mean, We can discuss about this offline if you're interested.
Of course.
Cool. Thank you.
Thanks.
Cool. We have 2 more questions from Hu. Thrombin has multiple receptors, therefore, upon inhibition of thrombin, GPIB and PAR1 / PAR4 complex will also be affected. Do you suggest that GRAP or the release of TGF beta works behind all these receptors?
Yeah, no, very nice question. I was surprised that the reviewer didn't give us a hard time with this question, and when we submit the work to STM. But this is a very complicated question. The way we get away with it is to use a GAB knockout animal. So we found that GAB knockout animal failed to respond to the thrombin inhibitor. So we then therefore, by deduction, stated that maybe GAB is the only thing. But of course, from your question, you can see we cannot categorically rule out the contribution by other receptors. So even though we said in the paper that seems like we know the answer, but in the back of my mind, I think the question has not been answered completely. So thanks for that question.
And one more question from Hu. Aspirin inhibits the production of thrombix A2. Clopidogrel inhibits P2Y12 receptor.
Mm-hmm.
Well, another drug inhibits thrombin. Those agents inhibit completely different pathways. How come that all those agents seem to have the same function in your anti-tumor assay?
Yeah, so I think all boils down to inhibition of platelet function. So I guess for somebody like me who don't study platelet biology necessarily, who you from your question, I know you are expert in this area. We sort of just attribute to just reduce the function of platelets. Therefore, that results in attenuation of the suppressive function of T cells. Of course, we think there is a common denominator, which is TGF beta. So all those inhibitors appear to reduce TGF beta. So therefore, I guess once again, just like your other question regarding GARB, contribution of GARB in the thrombin experiment, we cannot say that all those things, all the receptor leads to the same thing, right? Obviously, the pathway is very different, but all of which leads to platelet activation. So if you inhibit each of those pathways individually, you somehow dial down the platelet activation. You dial down maybe the ability of platelet to adhere, to aggregate, and to produce TGF beta. So therefore, collectively, all of which seem to leads to a better anti-tumor immunity. So I think that will be our explanation. But of course, devils are always in the details. So we need to do more experiments for sure.
Cool, perfect. I think if there's no more questions, let's thank Zihai again for wonderful talk and also wonderful questions for all the answers for all the questions. Thank you again, and hope to see you guys next week. Zihai, enjoy the rest of the week.
Well, thanks to everybody, and be safe, everyone.
All right, bye.
Take care. Bye-bye.